Dr. Emily Mitchell, thanks so much for coming on to Evolution Soup from your office in Cambridge, right here in the UK. You are an assistant professor at Cambridge University, researching the ecology and evolution of early animals from the ancient Ediacaran period, as well as similar communities of the Antarctic. We'll be talking about all that today, as well as your study of the evolution of life on other planets, and how the evolution of life here could very well reflect the evolution of life out there. Welcome to the show, Emily. We've covered the ancient eras like the Ediacaran before in this show, but never really touched upon life on other worlds. So when you mention your research to people, what kind of reactions do you tend to get?
So the reactions to my work is very, very varied. I mean, people don't really think too much about how understanding life here on Earth can be used to understand life on other planets. And so that's something that's really, really interesting to them. The problem really with trying to understand life on other planets is we only have one example of life as we know it at the moment, which is here on Earth. And we're only really dealing with one point in time if we look at life around us today. So if you really want to understand how we got here, we have to go back in time and look at the fossil record. And so what I'm really interested in is understanding how animals have evolved right from their first origination 600 odd million years ago to now, but also what that might look like if we had a similar planet elsewhere or indeed a different planet. If we're on a water world, are we likely to get animals? Are we likely to just get microbes or no life at all? And so by looking at the fossil record and understanding the big macroevolutionary patterns, the idea is that we can start to think about what things might look like elsewhere and how we might go about detecting them.
Before we talk about the evolution of early animals on Earth and other worlds, let's just hear a little bit about your background. Emily, can you describe the academic road that led you to the work you're doing now?
So I've got a fairly unusual background for a biologist and paleontologist in that I actually started off doing physics and maths and then started getting really interested in using these mathematics and physics skills to understand how ecosystems function. And so before doing a PhD in paleontology and earth sciences and then coming to the Department of Zoology in 2019, one of the really fantastic things about paleontology is in order to understand the fossils, you can use a huge amount of different techniques. So these are biology, earth science, chemistry, and indeed biology. And so by combining all these different disciplines, it's a really great subject to be utilizing all these interdisciplinary skills.
Well, we've dived into the weird and wonderful worlds of the ancient Cambrian and Ediacaran periods of Earth's history before in this channel, examining the very earliest of animals that evolved over 500 million years ago. Emily, can you give us an overview of your work, the locations that you study, and how this research can help us understand how life might have evolved on other planets?
So my work focuses on the very first of the Ediacaran animals, which are known mainly from Newfoundland, Canada, and Charnwood Forest here in the UK. So Charnwood Forest is where we first found large, complex fossils in the 1950s, and they were actually found by school children. So the first person to find them was a schoolgirl called Tina Negus. However, no one believed her because everyone knew at that time, or thought they knew, that it wasn't possible to find fossils in rocks that were Precambrian. The following year, a schoolboy called Roger Mason found them, told his school teacher and his father, and they got in contact with geologists, and that's when the first fossils were published from Precambrian rocks from Charnwood Forest. Unfortunately, Tina was ignored, both because of her social class and because she was a woman. But now things are much, much better in Ediacaran. We have a huge variety of different people working on fossil science across the world. Most of my work involves working in Newfoundland, in Canada, on the coasts. And this is a really fantastic place to work because you have thousands upon thousands of fossils literally on the rock surfaces, on the coastline. You can go out and then you can look at them and try and understand life at this time. So the preservation of these Ediacaran fossils is really, really exceptional. They're all preserved under volcanic ash flows. So it's a bit like Pompeii in that life at that time was killed very instantly and preserved where it was living. So the earliest of the Ediacaran fossils from Newfoundland are known as the Avalon assemblage, also from Charnwood Forest in the UK. And these Avalonian fossils predate the ability to move around, which means they're stuck to the surface of the sea floor. Now that means that when you kill them with volcanic ash flows underwater, you have preserved the entire community of life at that time. So a snapshot and a little window into Ediacaran life. Now this is fantastic, but it means that we can go out and we can actually see what's going on with these very, very early animals if we can capture the data from the fossil surfaces. Now this isn't as easy as you might expect, because the fossils are fairly shallow. They don't have much relief on the rock surface, which means they're hard to see unless the light's perfect. And unfortunately in Newfoundland, the light is very rarely perfect. At best case scenario, it's for half an hour a day, but most of the time it's incredibly foggy, which makes it hard to see the fossils. Also, if we're wanting to capture thousands of fossils over hundreds of square meters, we need to have a process that enables the resolution and the accuracy to be preserved on these sorts of spatial scales. And so for that reason, me and my group have been using laser scanning techniques which enable us to capture these huge bedding planes down to a micron level resolution while we're in the field, and then bring them back to Cambridge, the digital scans that is, not the fossils, and then analyse what's going on with them. So what's really, really cool about these fossils is because we have these snapshots or censuses of Ediacaran life, we can use a whole host of techniques that are normally not really applicable to the fossil record in order to understand what's going on. And so that's what we've been doing, using a whole host of quantitative, mathematical, statistical techniques to understand how these organisms are interacting with each other and with their local environment. You can also understand how they're reproducing, and this is because when it comes to organisms that couldn't move around, that were immobile, a little bit like trees or corals, there are only a limited number of processes that can impact them. And so these are things like how they reproduced, so how they got there, and how they're competing or interacting with their neighbours, and how they're interacting with the local environment. So we can use all these different techniques that are very well established in modern data to understand what's driving the ecological dynamics of these communities. However, in order to do that, we also want to be able to compare to modern communities where we can collect more data. So we can't make precise environmental measures for the Ediacaran. We've only got the rocks as they're preserved. Whereas in the modern, we can measure all sorts of different environmental parameters. And so that means we do lots of work on the modern in the deep sea, in tropical coral reefs, and also in Antarctica. So we use similar techniques in that we use these quantitative ecological spatial processes to understand what's driving the dynamics of these marine systems. In terms of the data we're using, again, it's unfortunately not collected by us when it comes to Antarctica, but we've got a huge amount of underwater vehicles, so remotely operated vehicles that can go and take out huge amounts of video of the seafloor, which we can come back, sent back to us. We make 3D models of the seafloor and then can analyse all the different positions of the different organisms on the seafloor to work out what the underlying processes are. So one of the biggest questions for my research is, why did animals evolve when they did? So life has existed on Earth for 4 billion years or so, but it's only around 600 million years ago that we start getting animals. Before the Ediacaran, life was just microbial. And it's really not clear whether it took a long time to build up the biological complexity that enables animals to evolve, or we just got really, really lucky and it's a very unusual thing to happen, or perhaps that we were actually really bad at it, and actually in other circumstances we might have got animals evolving much, much quicker. And this is why I'm interested in understanding what life might exist like on other planets outside the Earth. Because on Earth we only have n equals 1, we have no idea about these origins of animals. But if we can find other planets and get a sense of the likelihood that animals exist on them, then we can start making comparisons and actually understand the nature of evolution and complex evolution in terms of animals much, much better.
Emily, can you go into more detail on your studies of Antarctica, its biosystems, and why it's so helpful to your work?
So I first got interested in Antarctica because it presents a really nice analogue for the Ediacaran. So Antarctica is relatively pristine in terms of human impact. It's one of the few places on Earth where humans have a relatively limited impact, which is fantastic if you're trying to understand these ecosystems. But it's also really interesting because unlike everywhere else in the world, there really aren't predators that can crush shells and crabs and other sorts of big predators like sharks. So similar to the Ediacaran, there aren't predators swimming around in the oceans, which means that Antarctica is a really great analogue. And so I work with colleagues in British Antarctic Survey, and these colleagues will go down to Antarctica and they'll use underwater vehicles to collect loads and loads of video footage of the seafloor, which they then bring back to Cambridge, and we can then analyse what's going on with the seafloor dynamics. But it's also really interesting in terms of thinking about life elsewhere. So as you may know, in the solar system, we have a few icy moons around planets like Saturn and Jupiter, like Enceladus, which are totally ice worlds. But underneath, we know from the astrophysics done over the last few years that they've got hydrothermal vents. And this presents a really intriguing question that we might actually have the possibility for life existing underneath these icy moons right here in the solar system. So that what that means is Antarctica provides another really interesting analogue for trying to understand what life might look like elsewhere, because we could have these ice-covered worlds with life underneath them, potentially quite different in terms of what our modern oceans look like, but similar enough to potentially use the information we can gain from understanding Antarctica to start to look for life on these icy moons.
You mentioned that you used a very advanced type of scanning tech to digitally capture Ediacaran fossils. So, how does this technology work exactly, and how does it help researchers like yourself?
So these wonderful fossils are preserved on these very large fossil surfaces, but they need to stay where they were for two reasons. First is conservation. It's really, if you just take one or two fossils out, you run the risk of breaking all of them, and you're also taking away the context in which they actually lived. But also it's quite difficult to actually get them out of the rocks, and not practical. You couldn't take hundreds of square meters of rock to a museum. So what that means is that the fossils stay where they are, and so we need to capture the information from them digitally. Some people can use casts and molds, and we've done a little bit of casting and molding to capture these fossil surfaces. But that can be difficult in terms of very fine scale resolution because you start getting air bubbles. So what we've been using instead is a laser scanner. So this presented quite a difficult problem because normally when you're laser scanning, you either want to have a huge amounts of area, so hundreds of square meters, but at a low resolution, or you want to have very precise high resolution on small scales. And we wanted to do both. And what we found was a really good way to approach it was using a sort of scanner. It's made by Faro. The current one is a quantum scanner. And these were developed not at all for using any sort of science or paleontology, but instead for looking at aerospace and manufacturing. The idea was these scanners, because they're so high resolution, the most recent ones are down to 0.015 of a millimeter, so really tiny resolution. These are used to scan machines to try and find cracks or flaws, and if needed, they can then be used to reverse engineer the broken pieces. So these are normally used inside in terms of often clean rooms, but we decided to turn it on its head and take them outside. And this was amazing because it meant that what we could do is get this really high resolution that we need in order to ID the fossils and capture where they were on the fossil surfaces over these very large areas. So these Ediacaran life forms, they couldn't move around, they predate the ability to move, which means that where they are on the rock and how big they are encapsulates their entire life history. So that's how they reproduced, how they competed and interacted with each other, and how they interacted with their local environment. And because this is all encapsulated by where they sit on the surface, If you look at how the densities change over the surface and compare them to known processes today, you can actually quantitatively infer the probability that different processes contributed to the different community dynamics, including how they reproduce. So if you're reproducing like corals do by spawning, you have a huge amount of offspring that get carried by the water, and they kind of end up all over the place. And so you see some features of this. You see very, very large reproductive clusters, and you also see kind of current-born clusters, in that when you've got clusters where the offspring have been carried by the current, they will be kind of drawn out oval-shaped clusters, because you're more likely to get offspring going downstream than you are to either side. Whereas if you're reproducing in a different way, so for example, asexually, using filaments or stolons, in the way that strawberry plants or spider plants reproduce, then they stay very close to the parent because they're tethered. So you get very small reproductive clusters and very round ones. And so you can compare different sorts of reproductive mode by looking at these densities and comparing them to the known processes today. Another aspect of why we use the laser scanners rather than photographs, for example, is that to see the fossils is fantastic and relatively easy when the sun's good, but this only occurs for about half an hour each day, because the fossils are very low relief on the bedding surface, so you have to have the light hitting them exactly right in order to be able to see them. But most of the time in Newfoundland it's incredibly foggy. Indeed, in our, I think, most unfortunate field season, in a course of 4 weeks, we only had 2 days where we had good sunlight. So that was a total of 1 hour of sunlight. So we can't rely on photographing the fossils in order to be able to capture them, which is another reason why we're using the laser scanners. And so when we've got the laser scans of fossils, we spend about an hour to get about a metre squared, and then we stitch them all together. And in the end, we end up with hundreds of square metres. In total, we have about 934 square metres of Ediacaran fossil surfaces that we've scanned to date.
Your work also asks the big questions about life elsewhere in the universe. Emily, how does your research here on Earth relate to the evolution of life on other worlds?
So my research at its core is trying to understand what's driving the evolution of life, and the extent to which the patterns we see here on Earth are fundamental rules of evolution or may vary elsewhere. So in order to understand what life elsewhere may look like, we need to understand what we might be expecting. And to do that, we need to think about what's driving the big evolutionary transitions of life on Earth, of which animals is, for me at least, the most important one. If we find life elsewhere and it's a whole bunch of microbes, will we get excited? Well, lots of people will, but a lot of people really are looking for animal style and even human level life elsewhere. So in order to do that, we need a huge amount of technology and scientists. And so I work with colleagues in the Institute of Astronomy here in Cambridge, whose work focuses on looking at exoplanet atmospheres. Now, this is a really, really interesting area of research, because by looking at the atmospheres, you can start to work out what's inside them. So this is using telescopes like the James Webb Telescope. And so the idea with James Webb is that they get pointed at particular targets, exoplanets, and then by looking at how the light from their nearby stars changes as it goes through the atmosphere, you can see differences in the atmospheric spectrum, is what they're called, depending on what's in the atmosphere. And so you see peaks and troughs depending on different molecules. And there's a whole bank of different known spectra for different molecules. So what people do is they fit different models to the exoplanet atmospheric spectra to see what the different peaks and troughs are most likely to correspond. And that can then lead to understanding what the different molecules are present in different atmospheres. So a couple of years ago, a colleague, Nicola Madison, an astronomer, he's been focusing on Hycean exoplanets. So these are planets that have the hydrogen-rich water worlds, sub-Neptune water worlds. And so these are really, really interesting because Madhu and his colleagues managed to find very, very high levels of methane and carbon dioxide in the atmospheres. He also, more recently, managed to find signals that seem to be something called dimethyl sulfide, or DMS. Now, DMS is very interesting because on Earth, it's only produced by phytoplankton. That is, it's only produced by life. And so it hints at the possibility of life on other exoplanets. However, when you've got different atmospheres, you need to be really, really sure that what's going on is indeed life, not some weird chemical reaction that we're not aware of here on Earth. And so that's what a lot of people are doing at the moment, is trying to work out, when we see DMS, could it be caused by photochemical reactions, for example, and so abiotically. And so this is the first, and hopefully a series of different atmospheres that are being resolved on different exoplanets well outside the solar system. And if we start getting these different signatures, we have the potential to then work out, well, what sort of life is it? Is it phytoplankton? Is it more complex? Because animals, things like plants, things like oxygen producing phytoplankton all leave very distinct atmospheric signatures. So we can start to tie down what exactly is going on these planets in terms of life and interactions between the life and possible life and the planet itself.
Well, it certainly is incredible work that you're doing, and I'm very grateful that you've been able to take the time to come onto the show today. So, what's next for you? What new projects can you tell us about?
Well, it's been fantastic talking to you. So I've got some wonderful fieldwork coming up. We're going back to Newfoundland in a week to look at the fossils there and try and get some more data from there. And the other fieldwork we've got coming up soon is going to Fiji. So Fiji is really, really interesting from a coral perspective because there are loads of soft corals there. So most corals, we think of the hard corals from the coral reefs, but soft corals are really, really interesting because they're a lot more robust to climate change because they need less energy from the symbionts that the hard corals do. So sometimes when you have coral death and coral bleaching, the soft corals will kind of take over where the hard corals used to be. But we don't really understand this very well. Lots of people think about algae taking over, which is a lot more common. And so what we're doing in Fiji is we're monitoring areas that have a mix of hard and soft coral to see how they're changing over time and seeing how this then changes the dynamics of the local communities and their diversity. And this is interesting in terms of ediacarans because ediacarans are in many ways very similar to soft corals, but then everything changes once the animals grew the ability to, or evolved the ability to build hard shells like the coral reefs. And so this is another kind of aspect that we're just starting to work on, but a lot more of a long-term project, monitoring over the next 5 to 10 years.
Emily Mitchell, thank you very much indeed.
Thanks, Mark. It's been really great to chat to you about all this wonderful research.